Hi everyone, so we are here at your chapter 5 video. So if I am not mistaken, this is the last item that would be included on this week's quiz. So lipids would be your final item in this unit, and I think that's what I have listed on the syllabus. However, I think chapter 5 is where we're going to cut it off for the quiz content. So what is your quiz going to cover? Just as a quick review, quiz is going to cover chapters 1, 2, 3, 4, and then of course today, chapter 5. Remember that chapters 1 and 2 were largely introductory chapters. Chapter 1 was all about the study of biology and what constitutes life. Chapter 2 was an intro to chemistry with a particular focus on the chemistry of water and carbon as the foundational chemistry for biology. And then chapters 3, 4, and 5 were all part of our unit in macromolecules. It will also include chapter 6, but remember that's not on the quiz. So those are all part of our macromolecule unit. So today we're going to focus on carbohydrates. If you are keeping up, then you should have already watched one video on proteins, which is one type of macromolecule, and then one video on, oops, I'm just going to put nucleic acids. I almost put DNA. Nucleic acids, right? And in each case, we determined that these macromolecules represented polymers, so large molecules made up of repeating individual units. And the monomers for each of these polymers were amino acids, if you're talking about proteins, and nucleotides, if you're talking about nucleic acids. So today we're going to shift gears. We're going to talk about carbohydrates, which is another type of polymer made up of individual repeating units, repeating monomers called monosaccharides. All right. All right, so what do I want you to know from this chapter? All right, key concepts. I want you to understand the general structure of a monosaccharide monomer that might be used to make a sugar polymer. And I want us to understand how these monosaccharides link to form polysaccharides, which is our name for the sugar polymers, right? And you can probably already determine that saccharide is a suffix that we give to sugar molecules. And then pretty obviously we have mono or poly as our prefixes, meaning single or many, right? So this is our monomer, this is our polymer. All right, then finally, I want us to understand something about what carbohydrates do. So we're going to learn that there's three basic functions that carbohydrates can carry out within a cell. They function to add structure to cells. They help cells recognize one another as self or non-self. And they're also involved in cell signaling. So we're going to talk about that as well. All right, to start, let's talk about our sugar monomers. I referred to those earlier as monosaccharides. And hopefully we've seen this word enough times that I can just abbreviate, okay? So our sugar monomers are monosaccharides, but you've also heard me use this vocabulary term carbohydrate. So those seem like really different terms. Where does the term carbohydrate come from? Well, if you actually break down the term carbohydrate, right? You can see that it's really composed of two parts, carbo for carbon, and then of course hydrate for water. So if you were to look at the chemical formula for a carbohydrate, it would look something like this, CH2O to the N, where N is the number of carbons in the group, but you would multiply it times each of these, just like in math. So carbon, water, carbohydrate. So if you were talking about glucose, this N would be 6, and it would look like this, the chemical formula, C6H12O. H12O6. Does that make sense? So that's where that name carbohydrate comes from, okay? Now this N can be as few as 3, okay? Or as many as 1,000. And when we get up into the thousands, we're really into the polymers. The low numbers are usually for our monosaccharide monomers, okay? Now, let me just draw you sort of a very, very simple example of a 3-carbon carbohydrate monosaccharide monomer. So let's draw 3 carbons. And I'm just drawing them kind of like this to kind of give us some room. One of the things that we find is that there is a double bond in a carbohydrate between carbon and oxygen. And so we're going to see a couple of oxygen atoms in here, and that should indicate to you something. These are going to be polar molecules, right? Because remember that oxygen is highly electronegative. So if they're polar, that means they're going to be water soluble. Okay, so let me finish drawing this out. Let's see here, that's right, that's going to be an H, that's going to be an OH. Look, there's another oxygen, there's an H, and I think this one is an OH, H, and H. Now you can see that you don't have CH2O on each carbon atom, but if you were to count up the C's and the H's and the O's, it does work out to be a CH2O configuration. All right, so this is a very good example of what a carbohydrate monomer looks like. All right, in other words, a monosaccharide. Now you're going to hear some terms probably like as you move through biology and in your day-to-day life. So you've probably heard of monosaccharides before. These are simple sugars, right? Individual units that can be linked to form larger molecules. A great example of that is glucose, okay? You've probably heard the term disaccharide. Now, if monosaccharide means 1, then disaccharide means 2. Suffix saccharide equals sugar. And so if a monosaccharide is a simple sugar made up of 1 unit, a disaccharide must be a sugar made up of 2 units. Disaccharides belong to a larger group that we call oligo. Oops, that's an O. Oligo saccharides. Am I spelling that? Yeah. Saccharides. Ooh, spelling is my nemesis, y'all. All right. Oligosaccharides. The prefix oligo means few, and the suffix saccharides, of course, means sugar. So anything that has from 2 to 20 monosaccharides in it, we're going to call an oligosaccharide. But we often talk about disaccharides frequently because they're really important in our nutrition. We see them a lot. For example, sucrose is a disaccharide made up of glucose. Can you see that right there? And another 6-carbon sugar that comes from fruit called fructose. So this is common table sugar. This is what you put in your coffee, which I am drinking right now, okay? Reminded me to take a sip of my coffee. One more thing I wanted to point out. Look at this suffix here, ose. When we name sugars, when we give them their particular names, if they're going to have the suffix ose. So if you see a weird word, but it's got this ose suffix in it, you know that it's a carbohydrate, okay? You may not know whether it's a monosaccharide, disaccharide, or polysaccharide, but if this ose is in the name, you do know that it's a sugar. All right, finally, let's take a look at a real long polymer. Amylose is a polysaccharide. It's actually what we commonly refer to as starch. And if you look closely, what you can see here is that we have a series of glucose molecules linked together here. And this would go on and become a very long chain, okay? So from this slide, things I want you to take with you. I want you to know what a monosaccharide is, what a disaccharide is, what oligosaccharides are, what polysaccharides are, and I want you to understand that the suffix ose in the name of a molecule should indicate to you that you're dealing with a sugar. All right, now let's look a little more closely at sugar monomers, monosaccharides. So I've got for you here some 3-carbon sugars, okay? And I want to show you a couple of ways that carbohydrate monomers, monosaccharides, can vary among one another. Remember that if we're talking about proteins and amino acid monomers, they varied in their R groups, didn't they? And if we're talking about nucleotides, they can vary with the type of sugar that you have, right? You can have deoxyribose or ribose. And they could also vary with the type of base that they had. So they could have an A, C, T, or G if it was DNA, or an A, C, U, or G if it was RNA. So how do the monomers in sugars vary? Well, one way they can vary is the placement of that double bond that I showed you earlier. So sometimes you'll find the double bond at the end of the molecule. In that case, it would be called an aldose. I'm not going to test you on that terminology, okay? Or sometimes the double bond can be interior to the molecule. In that case, it would be called a ketose. Again, not really going to test you on that terminology. What I want you to understand is that one way that monosaccharides can vary is in the placement of the carbon double bond, okay? Of CO bond. Okay, see how that's a double bond? All right, so that's one way that we can have monomers be different from one another. Oh, okay, I was like thinking I had done the same slide twice, but this is a different slide. In this slide, how many carbons do we have? We have 1, 2, 3. Oops, in this slide, 1, 2, 3, 4, 5, 6. So another way monosaccharides can differ from one another is in the number of carbons, right? You can have some monosaccharides that are 3. You can have some monosaccharides that are 6. You can have some monosaccharides that are 5 or 4. For example, the sugar in the nucleotides that we just talked about, ribose and deoxyribose, they are 5 carbon sugars, okay? So for example, if you have a 5 carbon sugar, that is called a pentose. Like a pentagon has 5 sides, pentose. If you have a 6 carbon sugar, that is called a hexose. Okay? And let's see here, what else did I want you to know? Okay. The other way that we can have some variation, I want you to kind of hone in here on this OH group. And I like the way it's placed for you here. Do you see how this OH group is sticking up and this OH group is sticking down? So another variation is in the position of hydroxyl groups. Now you might think this is a little nitpicky, whether this is sticking up or this is sticking down, but I want you to take a very close look at the chemical models I have for you here. They look pretty doggone similar. The only difference is that this thumb is up and this thumb is down. And when we get that different orientation, we actually have different sugars. Glucose has this up orientation, galactose has this down orientation. So they're actually different sugars. And that means that they can be used differently, okay? These have different functions. You can eat galactose, right? But you can't really use it for fuel in your body until your body performs a chemical reaction using some enzymes that swaps this position here. Then you can use it for fuel, whereas glucose is all ready to go. So the reason that this placement is important is that your body recognizes the shape of molecules for enzymatic reactions. When the shape starts to differ, then your body starts to recognize these things as different and they have to be treated different in bodily functions, okay? So let's recap. I have given you 3 ways, 1, 2, 3 ways that sugar monomers can differ from one another, or monosaccharides can differ from one another. They can differ in where the double bond occurs. They can differ in how many carbons they're made up of, and they can differ in the orientation of their hydroxyl groups. All right? So that's 3 ways that you can have variation in monosaccharides. Now let's hone in on some of these longer carbon monomers, okay? These longer carbon monosaccharides, things like pentoses and hexoses. If you remember from when I was drawing your nucleotides for you, I was drawing that sugar as like a pentagon, wasn't I? With the oxygen in the middle. So let's take a look at that. What we find is that some of these sugars that have longer carbon backbones, they tend to form rings, particularly when they are in water solutions. So you rarely find glucose as a linear molecule. What happens more often is that this molecule folds in on itself, and you can see what's happening here. It's kind of folding in. Here's the top, here's the bottom. There's the top, there's the bottom. And what will happen is that this will lose this hydrogen, and this oxygen will connect here, and you end up with something that looks like that, okay? So we rarely find 5 and 6 carbon sugars in particular in linear form. They're almost always in ring form, at least in aquatic solutions, all right? Now, one thing I wanted to point out for you here is when this thing collapses in on itself, we have another situation with a thumbs down or a thumbs up for our OH group. And I told you that the positioning of that thumb, that OH thumb, matters for how your body is going to be able to treat these molecules. So if the thumb is down, we call that alpha glucose, and if the thumb is up, we call it beta glucose. I'm not going to ask you to remember alpha from beta glucose, but I do want you to remember that the position of that hydroxyl group does become important. And why is that, you say? Well, let's look here. If we have 2 alpha glucose molecules linking up with one another to form a chain, can you see that we have a V shape, right? If you have a beta and a beta, you end up with an A shape. So you end up with differently shaped bonds once you start linking these things together. And what that means is different enzymes are going to be able to break these apart to use that glucose for fuel later. So again, just that theme of the thumbs up versus the thumbs down matters for function in your body because it changes the overall shape of the molecule. And therefore, different enzymes are going to be the ones to attack it, so to speak. All right, now let's look a little more closely at how these things are linking up. So basically, we're going to see for polymerization, we have a monosaccharide here, a monosaccharide here, and what's going to happen, it's the same theme that we've seen again, is that we're going to lose a water molecule and we're going to get a linkage, right? We're going to get a bond. So once again, we're seeing that condensation reaction. Now, the bonds in proteins were called peptide bonds. The bonds in nucleic acids were called phosphodiester bonds, and the bonds in carbohydrates are called glycosidic linkages. Okay, so you can see here that we have a glycosidic linkage that's shaped like a V. Here we have a glycosidic linkage that's shaped like an A, right? And as I mentioned, it gets to be a little difficult for enzymes to manage some of these. Let me give you an example. So this is maltose. Maltose is made up of 2 glucose monomers, right? Glucose, and we have this nice little V. Maltose is very easy for your body to break down. Now let's take a look at this one. Remember I told you earlier that galactose was just like glucose, but it's got a thumbs up and glucose has a thumbs down. So what happens after our condensation reaction is you have this right here. Well, you have to have a special enzyme. This results in a sugar called lactose, right? galactose plus glucose equals lactose. You probably know from general knowledge that lactose is the sugar that you find in dairy products. And you may know that in order to digest lactose, you need an enzyme called lactase to break these apart. Why would you break these apart? Well, because you want to get at the energy stored in a glucose molecule, and eventually you would convert galactose to glucose and use it for energy. The problem is, many folks stop making lactase as they age, right? That is an adaptation for our young when they drink milk, but then once they stop drinking milk, then they don't need this lactase enzyme anymore and your body stops producing it. And that results in a condition that many of you have heard of called lactose intolerance. All right, lactose intolerance is when you can't break this down. And so, like, for example, I am lactose intolerant. I can't eat dairy products, which is a shame because cheese is freaking delightful. But what does that mean? Well, while I can't use this, the bacteria in my gut can, and they start going crazy, and then I feel very poorly. So that's an example of how these different glycosidic linkages that result from this condensation reaction can have very different uses in terms of enzymes. Now, if this were going to become a longer and longer chain, you would just continue to add to that chain with more and more glycosidic linkages to make longer and longer polysaccharides. All right. So let's see here. I think I've talked about all of these things. I do have notes for you down here. You can read those and review those and make sure you understand everything on this slide. All right. Let's look at some examples of some larger polysaccharide molecules that result from multiple glycosidic linkages, okay? So here's an example. Starch is one example. It's used for energy storage in plant cells, like in potatoes, okay? And what you have is a whole bunch of glucose molecules linked to one another, okay? And it's just showing you there's different ways they can link up. You can have a linear starch. Sometimes you can have branched starches, and those become something called amylopectin, but it's basically a polysaccharide resulting from multiple glycosidic linkages. Here's another molecule you may have heard of, glycogen. Glycogen is how we store glucose in our bodies. We store it for long-term energy use, and you often find your glycogen is stored in your liver. So like if you run a marathon, you use up all the stored glucose in your liver, and then you just have to start eating sugar toward the end because you've used up all your glucose storage. And you can see what our glycogen looks like. It's a little different than the starch or the pectin that we find in plants, but it still looks kind of similar. It's kind of simple, few branches, but lots of glucose molecules linked together. Now let's look at another way polysaccharides can form. This is cellulose. Cellulose is another set of glycosidic linkages between beta glucoses. And I want you to notice it forms almost a mesh. And this kind of gets to what we're going to talk about in just a second. We're going to talk about the different functions that polysaccharides or carbohydrates can have. You know, here we've got energy storage. Remember the NRG is my abbreviation for energy. You got energy storage, that's one function. This is going to be a structural function. Can you see how this mesh might be structured? This mesh might be really strong, okay? This is the stuff that you find in cell walls of plants, and it's the stuff that makes up woody stems and like wood pulp and your paper and whatever. So this is a structural carbohydrate that's really strong because of the way these glucose molecules are linked to one another. They are forming a mesh-like structure. All right, let's look at another example. Okay, chitin. You may have heard of this before. This is the stuff that makes up exoskeletons in insects and sometimes in fungi. Some of their cell walls have chitin in them. Again, you can tell that this is a structural carbohydrate because it is a mesh-like structure. And then the last one I wanted to show you is peptidoglycan. This is used for structural support in bacterial cell walls. So you can see here that it's actually a mixture of both glucose molecules and some amino acids, so some very short chain protein type structures. So you have not just glycosidic linkages, but you also have some peptide bonds here. But in essence, you have a mesh-like structure. So that's going to be a very strong situation, okay? So what I want you to understand is that if you have a mesh-like structure, it's going to be structural support. If you have a more linear structure with maybe a few branches, it's best for energy storage. So if I were to show you a picture on a quiz and say, is this a structural polysaccharide, or is this polysaccharide used for energy storage, then you should be able to differentiate between the two, all right? So I kind of started talking about the functions a second ago, and I mentioned that one of the functions of carbohydrates is that they can be structural, right? They can provide structure. And I mentioned chitin as an example, and I also mentioned cellulose. I feel like we've got that. We know that that results from a mesh-like structure in the polysaccharides, right? So you've got cellulose. Remember, those are plant fibers, plant cell walls. Chitin, I've got for you here a really nice picture of an exoskeleton from a cicada. You guys have probably seen these before. And all of that would be made up of chitin, all right? Another really important function of carbohydrates is they can be used in cellular recognition. What do I mean by that? Well, sometimes your cells need to be able to recognize whether they come from you or whether they are foreign invaders. Or sometimes your cells need to know, oh, is that a liver cell or is that a brain cell? Is that a muscle cell or is that a bone cell? So the way that that happens is we use carbohydrates as tags to identify the type of cell, okay? So here I have for you something called a glycoprotein, okay? The suffix glyco, sorry, the prefix glyco means sugar, and then of course protein means protein, right? So you can guess that a glycoprotein is a protein that has a sugar tag on it. Notice this is labeled as an oligosaccharide, so it's got a few monosaccharides in it. Remember I said it's usually less than 20. 1, 2, 3, 4, 5, 6, 7, 8. So that fits the definition, doesn't it? Okay. Here is an example of a glycolipid. So lipids are fats, and you're going to see that they are also used in cell membranes. We're going to talk about that in chapter 6. But for now, I just want you to recognize that a glycolipid is going to be a lipid that has a sugar attached to it. So these sugars make great identification tags. Why is that? Well, most sugars are polymers that are made up of a couple monosaccharides, but some are combos of many different types of monosaccharides. And that allows us to have lots of different variety in what these sugar tags might look like. So you can have lots of unique oligosaccharides by just sticking together a bunch of different sugar monomers, and that allows you to have unique ID tags for your cells. I'll give you an example. Your ABO blood type is a result of different sugars on the surface of your blood cells. If you are type A, you have a particular sugar on the surface of your blood cells. If you're type B, you have a second type of sugar. If you are type O, you lack those sugars. And if you're type AB, you have both of those sugars, okay? And so that helps with recognition. All right. Now, one thing I wanted to show you, sometimes I like to show you guys how we come up with these things and kind of lead us through critical thinking exercises to show you different concepts. So I mentioned that there are lots of different situations where your body would want to be able to recognize whether they're your cells or they came from somewhere else, or to recognize different cell types. And one situation where it's important to be able to recognize different cell types is during fertilization. So, you know, sperm, as they are going through the act of fertilization, the way that they know when they have reached an egg is they recognize the cellular ID tag on the egg. Now, back before we understood that the ID tag on the egg was a carbohydrate, the way that researchers were able to figure that out, they basically took the proteins and the sugars and the glycoproteins that were on an egg. They took these glycoproteins and they said, wonder how these sperm are recognizing the egg. Are they recognizing the protein part of this molecule, or are they recognizing the sugar part of this molecule? Let me go back. So in the egg membrane, there are these proteins and these sugars, right? And they knew that's what the sperm were recognizing, but they didn't know if they were specifically recognizing this as the ID tag, or were they recognizing this as the ID tag? So they performed a very clever experiment. They were able, through the magic of science, to disrupt this molecule and separate it into its component parts, where you have some carbohydrates over here, all by themselves, these, and then you have the proteins over here, all by themselves, so just this part. Then what they did is they mixed a bunch of sperm in a petri dish with each of these fractions, with just the carbohydrate or with just the protein, okay? Now, what that means is once the sperm recognizes the correct ID tag, it's gonna stop seeking out an egg. Okay, so what should happen is when you introduce an egg, there's going to be what they call attachment inhibition, because the sperm should have already recognized the ID tag in the petri dish. When you add an egg to the petri dish, these sperm are already occupied and you should have attachment inhibition. So to figure out which fraction contains your cellular ID tag, you measure the one that has the most attachment inhibition. Now let's look at their results. So they took the intact glycoprotein, okay, the whole thing, put it in a petri dish, let the sperm interact with it, then plopped an egg in there, and they measured the attachment inhibition. And you can see that it was pretty high when you have your intact glycoprotein, okay? And then they had a control group where they didn't have any glycoproteins in it, and the sperm attached to the egg really well, okay? There was no attachment inhibition. So if you add the glycoprotein, you get blocking of attachment. You don't add the glycoprotein, the sperm seek out the egg just fine. Now let's look at the results after they break this molecule up. If you add just the carbohydrate, you get a lot of attachment inhibition, or in other words, you're blocking the sperm from going and attaching to the egg. But here you get very little attachment inhibition. In other words, the sperm are finding the egg just right. So using this experimental setup, these researchers were able to determine that it's the carbohydrate that's acting as the cellular ID tag, okay? You should be able to explain this experiment to me and explain to me how they reached the conclusion that carbohydrates are functioning like a cellular ID tag, okay? All right, so we've gone through 2 functions, 1, structure, 2, cellular recognition. All right, last but not least, the most famous and probably most important function of carbohydrates when we're talking about cellular biology is energy storage, right? That's what you think of the most when you think of carbohydrates, right? You guys know that if you eat a bunch of sugar, you get a sugar rush. So basically, let's look at what's going on with energy storage. The energy in carbohydrates is stored in these bonds here, and I'll show you why in just a second. But let's just remember where we're getting that energy from. That energy is coming from carbon dioxide in the atmosphere that is being used by plants They are using the energy of sunlight to take these carbons, mingle them with water, and create these carbohydrate molecules for the purpose of energy storage. So right here, what I have for you is linear glucose, and then of course we know that it folds into a ring. So this is glucose. So plants are harnessing the energy of light using carbon dioxide to kind of create molecular bonds where they can store that light energy as chemical energy and use it later as their cells need it, all right? Now, why is it that carbohydrates make for good chemical storage, good chemical energy storage? Well, let's look here. Carbon dioxide, you can see, is a highly polar molecule. Remember me telling you that oxygen loves to hog all the electrons, doesn't it? And so you can see that oxygen hogging these electrons means that they're already kind of off to one side, and that's going to make this a relatively easy bond to break apart, right? You can just break it off because all the electrons are already kind of over here. Now let's look in contrast at what a carbohydrate looks like. Can you see how evenly spaced these electrons are between these carbon-carbon bonds and between these carbon-hydrogen bonds? They're nice and evenly spaced. It's going to be harder to break these apart, isn't it? Because they're kind of really sharing those electrons equally between one another. And so in that way, we are able to take that sunlight energy and store it as chemical energy in these high energy bonds, okay? High energy bonds. And just to review, it's easier to snap these off because the electrons are already pretty much associated with oxygen, right? Going to be a lot harder here because these electrons are really evenly shared between these guys. All right. And I think, yeah, that's the end of this particular chapter. This one was a short one. Okay. So this is the last chapter that you'll be responsible for on your quiz. If you have any questions, I'll be glad to talk to you about them during our class session meetings this week. Talk to you soon. Bye-bye.